By the way, another word for homozygous is purebred, and another word for homozygous is hybrid. Anybody ever have a purebred dog? A purebred dog is simply homozygous for a particular set of genes that code for its appearance. Mendel's 3 principles. Three principles of life from Gregor Mendel himself. Here are your 3 principles. First of all, we have the principle of dominance. This states that some genes, some alleles, he called these factors because he didn't know alleles yet. Some alleles are dominant over others. And by dominant, he meant they take precedence in their expression. So anytime you have a dominant allele, you have that particular trait being expressed no matter what. Okay? Secondly, Mendel came up with the law of segregation. You have 2 copies of any particular gene, one from mom and one from dad. During the formation of gametes, of sex cells, these 2 copies will be separated into separate cells such that each generation maintains the same amount of genetic information, right? It's not like your kids have double the genetic information and your grandkids will have double amount of genetic information. Pretty soon our cells will be overrun with DNA. Wouldn't that be true? And so according to the law of segregation, the 2 copies of any gene separate during gamete formation. In other words, when we are forming gametes, sex cells, each sex cell only inherits 50% of the genetic information because the 2 copies separated and only 1 copy will be given to each gamete. This ensures that each generation will have the same amount of genetic information. Okay? Lastly, Mendel developed the law of independent assortment. Which means that the inheritance, I'm going to add a caveat to this, the inheritance of unlinked genes are statistically independent events. What does it mean to be statistically independent? We all dead breaks today? Hedvig, what does it mean to be statistically independent?
Varying?
Well, it means that the outcome of one probability is independent of the outcome of another one. In other words, let's say we have this genotype, A, B, B. According to the law of independent assortment, the inheritance of A is independent of the inheritance of B. In other words, just because you got stuck with capital A doesn't necessarily mean that you will get stuck with capital B. The inheritance of A is independent of the inheritance of B. Does that make sense? And now we know that this is only partially true under certain circumstances. Does this sort of make sense? We've been learning this since like 7th grade, right? Do I need to go over Punnett squares? Oh. In a Punnett square, what happens is we line up the genotype on one axis and the other genotype of the other parent on the other axis. Okay? And so you can watch the god. You can find out the probabilities of each possible genotype. We call this a monohybrid cross. If I had two genes, we call it a dihybrid cross. Okay? Remember, in a monohybrid cross, we have a 3:1 phenotype ratio and a 1:2:1 genotype ratio. Think about what this means. What is a genotype? What is a genotype? Somebody remember what is a genotype? Yes?
The genetic makeup?
The actual genetic makeup. So this AA is the genotype. That's the actual set of alleles that this individual has. What is the phenotype? The physical appearance, right? So capital A, little a, capital A have the same phenotype but a different genotype. Now in a monohybrid cross, you're going to have a 3:1 phenotype ratio because 3 out of the 4 will have the dominant trait and 1 of the 4 will have the recessive trait. We have a 1:2:1 ratio for the genotype. Okay? Does that make sense? Okay. Now, it turns out that Mendel was working with a very simplistic system. Mendel's system of pea plants was incredibly well behaved. He just so happened to stumble across a system where every single gene was unlinked. And we'll talk about what it means to be a linked gene in a moment. But where everything obeyed a perfect dominant recessive pattern and where things are pretty much unlinked. Mendel worked with very nice data. Life is not so nice. Oh, that's a lot of dots. Okay, forget all that. But the idea here is that Mendel worked with very simplistic systems. There are certain extensions to Mendel that we need to be aware of. These are cases wherein simple dominant recessive inheritance patterns don't quite work. The first of these is incomplete dominance. In incomplete dominance, what happens is you have a mixing of the phenotypes in the heterozygote. Now, this stuff, a lot of the stuff is review. I want you to be familiar with articulating it, though. Just because it is review doesn't mean that it's not worth reviewing. As an AP biology student, you should be able to articulate all these things in relatively concise terms. Okay? So we all understand what dominance and recessive is, but sometimes we have a little bit of trouble in articulating what dominance actually means. Does that make sense? It's not a bad problem to have, but it's hard. So here are some extensions to Mendel. You might be down. The first one is incomplete dominance. In incomplete dominance, we have a mixing of phenotypes in the heterozygote. So for example, in carnations, crossing a red flower with a white flower will yield pink flowers. The heterozygous individual is actually pink. Okay? The reason why this is so is because this is actually, the type of dominance that something undergoes lies in the eyes of the beholder. And here's what I mean by that. If we look at the molecular level, this is technically a simple dominant recessive pattern. Because what happens is the dominant allele is making functional red protein, or functional red pigment. Okay? The white gene is producing no red pigment. So technically, at the molecular level, this is a simple dominant recessive system. But at the organismal level, when we zoom out, it appears pink because it produces less pigment when you only have one gene making it versus two. Yeah?
Isn't this the same as codominance though? Is this the same as codominance?
Yeah, so it lies in the eyes of the beholder. So in incomplete dominance, generally speaking, we're talking about large-scale organisms. Great. Okay. Nice. Cute. Okay. Any questions about incomplete dominance? In codominance, both traits are independently and distinctly represented. Two phenotypes are distinctly and independently represented. Okay? In codominance, we have each trait is independently and distinctly represented. There's no mixing. For example, if you had a black cow and a white cow, that's racist. A brown cow, that's even worse. A pink cow and a blue cow. A red cow and a blue cow. Republicans and Democrats, that's political. A red cow and a blue cow, and they were undergoing incomplete dominance, out comes a purple cow. But if it was codominance, then you'd have a splotchy red and blue cow. That sounds like something out of a nightmare. But blood type is a very good example of codominance. In blood type, you have three alleles. You have the A allele, which produces the A antigen. You have a B allele, which produces the B antigen. These are separate molecules that are being expressed on the surface of the cell. If you have both of these, both molecules will be expressed distinctly. And finally, you have the I, which produces no antigen. Okay? That's an example of codominance. So here are the different possible blood types and genotypes. Does anybody know your blood type? What's your blood type?
I'm like B, but my sister's like O, so.
Oh, so the two of you together are Bo.
Yeah.
So if you have type A blood, there are two possibilities. You might either be homozygous dominant or heterozygous. Both of those individuals will produce A antigens. Now, your body produces antibodies against the antigens it doesn't recognize. So if Montalban has type B blood, your body is producing anti-A antibodies because your body does not recognize the A antigen. And so if it sees it, it knows that it's coming from some other organism. Does that make sense? Does anybody else know your blood type?
I'm O.
You're O? You're not siblings, right? Good. Okay, that's weird. Sorry, that's wrong. Anybody else know your blood type? B. On your driver's license, it actually shows your blood type in case you like die in a car accident, or you're about to die in a car accident. More. No? I was thinking of blood tests. And I was part of a study, and the only reason why I did is because they paid very handsomely. I should not say this, huh?
How much do they pay?
A few hundred per hour of doing this study, of being part of this research study. And you're a research subject, but you have to get your blood tested every 3 months.
Wonderful.
It was a study on bone density in distance athletes. So they picked like 30 distance runners, a bunch of long distance swimmers, a bunch of bikers, and they looked for correlations between blood content and bone density. And they didn't find much. Anyways, but what they would do is they'd hook you up to a machine, and you had to go on a 48-hour fast, and you had to run as fast and as hard as you could until you passed out. And they had like a mattress behind you until you passed out. And they also took a blood test every 3 months. One day, I have low blood pressure. One day they took out too much blood too quickly and I passed out, or nearly passed out. And then I had to take a midterm half an hour later. Anyways, I don't know why that's related. Oh, that's related because I found out my blood type doing that blood test. Okay, type B blood likewise can be homozygous dominant or heterozygous, and you'll produce anti-A antibodies. If you have both A and B antigens being present, you're homozygous dominant for this. It's codominant. They're both being expressed. What kind of antibodies do you think you're going to make? You have both antigens, so your body is going to recognize both and not produce either of those antibodies. And if you have type O, like HEPA, their cells don't express either the A antigen or the B antigen, and so therefore you produce both anti-A and anti-B antibodies. Yeah.
What's the difference between like a positive and negative?
That's a different thing. That's the Rh factor.
I know. Wait, O, right?
Yeah.
Okay, so O negative
It's
is,
like the donor. or something.
yeah, it's a universal donor, and O positive is just normal, just can't take anything except O.
Oh.
Yeah, if you're type O blood, you're very philanthropic. You're a universal donor because you can donate your blood to anyone because it doesn't have any A or B antigens. Nobody's blood will get angry at it unless it has the Rh factor. If you're type AB blood and you get like a Caraxin, you're really lucky because you're the universal recipient. Your body doesn't produce either of the antibodies, and so you'll be able to receive blood from anybody with a close match. There's all other things that you look at when you do a blood transfusion. Okay? Anybody ever donate blood before? Huh? Oh, okay, never mind.
So if you like get tested for ACE, for human growth hormones, that extra blood, does it get donated or does it just go away?
No. You get tested. When you do a blood test on you, they don't like keep that blood going. They just toss it. Okay, check this out. Pleiotropy. In pleiotropy, what happens is one gene affects multiple characteristics. Okay? For example, I'm sorry?
Is this still an extension of Angola?
Yes, pleiotropy. This is when one gene affects many phenotypes. Okay, for example, the same genes that code for dwarfism also code for all sorts of skeletal system disorders. Okay? You have very large fingers, things like that. Wow, Andre the Giant. Wow, isn't he in like some movie?
Yeah.
Scary. Epistasis. This shows up a lot on AP Biology FRQs. In epistasis, one gene called the epistatic gene affects expression of another gene, the hypostatic gene. For example, if anybody have a Labrador at home, golden, brown, or black? Anybody have a Labrador at home? What? Your sister's not a Labrador.
My cousin has a half.
Oh. Your cousin's not a Labrador.
No, but she has a half.
So it turns out that there are only two genes that. Okay, I'm going to skip to this one. There's only two genes that code for fur color in Labradors. Okay? There's an epistatic gene, which we'll call E, and there's a hypostatic gene, which we'll call B. Now, normally in golden Labradors, or Labradors in general, okay, capital B makes black pigment. And this is dominant. Over little b, which codes for brown pigment. That's recessive. But whether or not you get black or brown Labrador is secondary to whether or not there's actually any pigment in the fur. The epistatic gene, E, capital E means that pigment is deposited in the fur. In other words, whatever pigment you're making will actually make it into the fur color. In the lowercase e, the recessive epistatic allele, no pigment is deposited. such that if I have a dog with this particular genotype, E, E, B, B, what color would this guy be? Pigment is displayed and it would be black. What about this one? Capital E, little b, little b.
Brown.
That would be brown. Because pigment's deposited and you get brown pigment. But what about this one? Little e, big B, little b.
Brown.
Golden. Because even though you're making black pigment, none of it gets deposited. So this gene affects whether or not this one will be affected. That's epistasis. Does that make sense?
So it's E, like small e, small
Anytime you get small e, no matter what this is, it's going to be golden. Because no matter if you're making black pigment, brown pigment, who cares, pink pigment, whatever, none of it's making it into the fur.
Oh, so aren't there like red golden retrievers?
No,
That's
it's
probably
just
some other
dye.
genetic anomaly. Or they have some hair dye. Anybody does research with Daniel? The weirdest hair ever. Who's ever seen Daniel? He's got like
You get that pink hair stripe down.
Daniel, if you're watching this video, don't be offended. Okay, any questions about this? Okay, so in pleiotropy you have one gene affecting many possible phenotypes. In a polygenic trait, you have something slightly different. You have many genes affecting one phenotype. A polygenic trait. A polygenic trait occurs when many genes affect one characteristic. And often when you have a particular trait coded for by polygenic inheritance, you'll end up with a bell curve distribution. A bell curve or a normal distribution. Take, for instance, the inheritance of different skin pigments. Look at this bell curve here. Some people in this room have really dark skin. I'm not being racist. Some people in this room have really light skin. Not being racist. But the distribution of people with really dark skin and really light skin is relatively minimal. Most people have something in the middle, right? We all have pretty intermediate skin. Yes? Okay. Does this sort of make sense? Many characteristics of humans are coded for by polygenic inheritance. Wow. Okay. Albinos. Scary.
For the guy that we're
More about albinos. Wow. Okay. Some genes are sex-linked, which means that they occur on the X and the Y chromosomes. Remember that if you have two X chromosomes, you're biologically what gender? Female, and if you have an X and a Y, you're biologically what gender? Male. Yeah.
So for transgender people, how does that work? So if you're born a guy,
Born gender.
how do you feel like a woman or the way around?
I'm not going to go too far into that because that will end up in a political discussion. But if you're transgender, you're born biologically one gender. Nobody's born biologically two genders at once. Yes.
Yeah, there was a condition where you have both. No, it can't.
I'm going to go over that in a little bit. So those are genetic anomalies. Those are not genders. Those are anomalies, right? If you have Turner syndrome, you just have one X chromosome.
It's weird.
If you have Klinefelter syndrome, it's XXY. Those are not genders. It's not like a Turner's gender. It's just an anomaly in the genetics. Yeah.
So if you're Turner syndrome, how does that work out?
You're a female with underdeveloped female characteristics. And if you are Klinefelter syndrome XXY, you're male with feminine characteristics. Okay. You can Google it if you're more interested in that stuff. So I'm not going to do all too much on that. Okay. Some genes are what we call autosomal. They exist on the regular chromosomes. Some exist on the sex chromosomes, and therefore we observe a different inheritance pattern. Okay, so sex-linked genes, I'm just going to put these in their own category. Sex-linked genes occur on the X or Y chromosomes. By the way, in some species, gender is determined not by chromosomes, it's determined by developmental temperature. And in things like, I believe, butterflies and other insects, the male is the homozygous one. So it's strange. Anyways. Remember that we have how many chromosomes? We have 23 pairs, 46 chromosomes. This is a karyotype. It is a picture of your chromosomes. And do you see that thing down there? Those two things? You have a huge X chromosome and a small Y chromosome. The X chromosome has a lot more genes than the Y chromosome. So most sex-linked traits are on the Y chromosome. There are only a few sex-linked traits which are coded for by genes on the Y chromosome. The X and Y chromosomes are not homologous. They're not homologous. They don't carry the same genes. Okay? Does that sort of make sense?
So most sex-linked traits are on the X chromosome or Y chromosome?
Most sex-linked traits are on the X chromosome just because there's more real estate there. Okay? Does that make sense? And this is in humans specifically. If you go to a different organism in a different phylum, this may or may not be true. Okay? Okay, the genetics of sex, we mean gender, whatever. Okay, XX is female, XY is male. Okay? In females, you have two X chromosomes, yes? Now, most cells don't need two copies of the same chromosome. One of them is going to get randomly shut off. Okay, so in females, you have redundancy. In males, you don't have redundancy. There you go. Look, there's 50%. Anybody have who is a girl with a brother? How many boys have a sister?
Wait, what?
How many boys have a sister? Yeah, so it turns out that most families end up having some kids from either gender. Some, I guess, get one sister. Must be interesting.
It's less fighting.
Ah. You have 3 brothers. Anyways. Does this sort of make sense? We're not going to talk about these slides. But the Y chromosome simply has fewer genes than the X chromosome. It is a master regulator for maleness. It turns on genes that are responsible for production of male hormones. By the way, do females have testosterone? Yes, just in different amounts. Do males have estrogen? Yes, just in different amounts. It's all about relative amounts. Okay. X chromosomes have a lot more genes beyond simply determining maleness or femaleness. Things like hemophilia, muscular dystrophy, color blindness, these are all X-linked genes. Okay, the X chromosome has more genes on it simply because it has more real estate. Yes, you can fit more people in the US than you can fit, actually you can fit more. That's a bad analogy. Forget that analogy. Okay. I didn't just compare the US to the X chromosome. It's okay. So here's a gene map of the X chromosome. Look at all those different sex-linked disorders that could occur. Look, Pelizaeus-Merzbacher disease, Fabry disease, immunodeficiency, sideroblasts. I don't even know what these things are. If you have questions about what these things are, ask Matthew because he's the med guy. On the Y chromosome, though, not so many. Not so many. Look, you can lose. Look, total lack of recall for date. Oops. Huh? Anybody like the names of these genes? Selective Curie loss. Well, I mean, the total lack of recall for dates. The oops gene.
Addiction to death.
Wait, I think I was self-conscious.
This is a joke. This is not real.
Itchy.
This is not real.
Oh, spinning is the true way at the end of it.
Okay. This is not real. Okay? There's no gene called air guitar.
Well, what's a self-conscious gene? It plays too.
Okay, anyways. Often, when you have a lot of inbreeding, a lot of incest, we'll call it incest, what happens is back in the 1800s and 1900s, many of the royal families of Europe were basically cousins and families of each other. Like the Queen of England was like the long-lost cousin, not a lot, like one generation separated from like the, her cousin was like the Vice Duchess of Prussia, I don't know. But everybody in Europe was sort of interrelated, and they only married royal blood. In other words, there were like 3 families, and they just like made babies with each other, and all those babies became kings and queens of everywhere. That causes a lot of genetic problems, because if there is a mutation in an X chromosome, or really any sort of recessive mutation, it's much more likely to get a homozygous recessive individual when you have a lot of inbreeding and a lot of incest like that. Does that sort of make sense? This is why purebred dogs run into a lot of genetic disorders, because as you inbreed a lot, basically there's like a group of like 10 dogs that are making babies with each other all day. You have an increased probability of inheriting 2 faulty genes. Right? I just was a purebred dog. Sorry. Okay. So hemophilia is a genetic disorder where individuals are unable to produce the proper clotting factors. Remember we talked about the clotting cascade? That ultimately ends in the conversion of profibrin to fibrin. It's a long cascade of signal transduction. If one of those is coded for by a gene on the X chromosome. And so individuals with the who inherit the X allele with that defective gene will become hemophiliacs, which means that they are unable to make that clotting factor. If they're unable to make that clotting factor, what do you think happens with them if they get a paper cut? They bleed to death. Now watch this. Inheriting an X-linked disease is like stepping on a landmine. X-linked dominant genes tend to be expressed more in females. Because in females, you have 2 copies of the X chromosome, and hence you have twice the likelihood of inheriting a bad copy. Does that make sense? X-linked dominant traits show up a lot in females. Most sex-linked disorders are, however, X-linked recessive. X-linked recessive traits tend to show up more in males because a male only inherits 1 X chromosome. In a female, you have the other one. If the other one's okay, then you're okay. But in males, if you get stuck with 1 bad copy, bummer. Which is why color blindness occurs several-fold times more frequently in males than females. Color blindness, hemophilia are disproportionately, they land on the side of males because males only inherit 1 X chromosome. And so you don't have the other X chromosome with the dominant allele that's normal to safeguard against the inheritance of a recessive allele that's defective. Does that make sense? Now we said earlier that females have 2 X chromosomes. Okay, there carrier E-screen. Okay, females have 2 X chromosomes. One of those 2 chromosomes is randomly inactivated during development. Okay, it gets inactivated, and so it is condensed into this heterochromatin lump called a Barr body. Remember, heterochromatin is when the chromosome is condensed and jam-packed together, and thus it is not being expressed. Now, for females, it's a coin flip. This happens sometime during development. And so whether you get the trait on one X chromosome or the other is completely random. This is how you get calico cats. One of these alleles codes for orange fur, the other one codes for black fur. And one of these X chromosomes carrying one of those alleles is randomly inactivated during development of the cat. And so you end up with calico cats when one of those ends up being inactivated. And so you see the cat over there, all the brown or all the orange fur all came from a cell once upon a time when the cat was in love with cells that deactivated the chromosome with the black gene. And all the black patches came from progenitor cells that randomly inactivated the chromosome with the orange gene. Does that make sense? Okay. Your textbook also has another more gross example, which I will not care to elaborate on. Any questions about Barr bodies? Yeah? No? Another question? So anytime you see a calico cat, a tricolored cat, it has to be a female, because that means that it has Barr body inactivation. Okay, one of the X chromosomes is inactivated into this heterochromatin lump called a Barr body. Okay? Now, my roommate in college had a receding hairline. Wow, that guy looks very intense. Okay. In males, because you only inherit one copy, you're more likely to end up bald. My roommate had a receding hairline. He used this cream called Bald X, trying to get his hair back. I told him one day, I think it's just making me balder by the day. How to gain influence and make them. I think he's bald. I don't know. Receding hairline. If he's got like hair all the way around here, but like this is all gone. That's not a very nice way. One time I told him, like, why don't you shave it all off? It's going to end up that way anyways. You can get over it. The same. Does this sort of make sense? So we're going to start to transition a little bit into the basis for molecular genetics. Ultimately, all this stuff is great, but it's very much so based on statistics and based on probabilities, yes? Based on macroskeletons. Let's talk about the nitty-gritties behind the biomolecules responsible for genetics. Ultimately, chromosomes are related to the phenotypes. T.H. Morgan, 1908, working with fruit flies. Anybody do research with Alicia? My goodness. She has fruit flies with genetic anomalies with no eyeballs. With tiny eyeballs. And so they like escape every once in a while, and they can't see where they're going, so they hit you in the head. They can't see because they don't have eyes. Anyways, Drosophila is a very heavily studied organism. You can buy them on Carolina Biological Supply for like $30. You too can have a little vial of eyeball-less fruit flies. If you want them as pets or something, I don't know. But T.H. Morgan was the first to associate the fact that they had a particular visible trait with the fact that they had a particular chromosome. Okay? Before this, it was really poorly understood as to how these traits were passed along. Frederick Griffith was able to transform bacteria. He determined that there's some sort of factor that can be transmitted from bacteria. I'm not going to go too much into this because we have other things to cover. But here's the important thing. today we understand that DNA is the molecule that transmits information, yes? DNA is the molecule that transmits information. Go back 100 years, that was not so obvious. There's a huge argument about whether it's DNA or whether it's protein that transmits information. If you are a scientist and you want to determine if it's DNA or protein, one way you can do this is you can radio tag. You can give a molecule a radioactive tag on it using an element that's only found in DNA or protein. What is an example of an element that's only found in DNA and not in proteins?
Phosphorus.
Phosphorus. And so we can tag DNA with radioactive phosphorus. What's an element that's only found in proteins and not in DNA? Sulfur. So we can tag proteins with radioactive sulfur. And so what we do here is we tag a virus with radioactive phosphorus and radioactive sulfur. And we see when the virus infects a cell, what part is radioactive. And Hershey and Chase demonstrated that it's the radioactive phosphorus that makes it into the bacteria cell in their DNA. And the radioactive sulfur is not transmitted to the cell. This means that the radioactive sulfur is not the information carrying molecule. Okay? Does this sort of make sense? Hershey and Chase, don't they look fun? People always think that Chase is another guy, but it's actually a lady.
What?
Oh, and that's like a massive misogyny you got there.
It's a massive misogyny.
What's a misogyny?
A misogynist.
Misogynist is somebody who hates.
Okay, no. I thought Chase was the guy there because I looked at.
It comes from the Greek roots miso, or miseo, which means to hate, and gune, which means woman. Sorry, that Greek words are not very fun to pronounce. Okay. Any questions about this sort of stuff? Going once, going twice. So, let's talk about linked genes. What does it mean when genes are linked? So check this out. Ultimately, and I'm not going to talk about meiosis today, by the way. You're still responsible for the material that's in the video. Meiosis is not too bad, right? Do I need to talk about PMAT?
What's what?
Meiosis? Would you guys like me to talk about meiosis? You've been doing mitosis, meiosis since like 7th grade, which is why I completely gloss over this stuff. Matthew is teaching his 7th graders meiosis, I believe, on Friday. So, okay. Check this out. If you think about Mendel's luckiness, let me open up my link gene slides. You have access to all these slides on Dropbox. There's 160 slides in this presentation. Here we are. Okay, so genes that are, don't confuse these two terms. A gene that is located on a sex chromosome can be said to be sex-linked. Genes located in other chromosomes, but when two genes are on the same chromosome, We call these linked genes. Different from sex linked. What we mean by linked genes is that two genes located on the same chromosome will tend to be inherited together. Two genes on the same chromosome will tend to be inherited together. In other words, they will not independently assort. We said that Mendel got very lucky. And here's what we mean by Mendel got lucky. He happened to pick a whole bunch of traits that happened to be on very different chromosomes, and he actually did pick two genes that were on the same chromosome, but they're very far apart. So that wasn't an issue for him. He never ran into this. But certain genes tend to be inherited together. We call this issue linked genes. I'll give you this example. Okay? Remember in meiosis, you have crossing over, yes? Now, in meiosis, in metaphase 1, remember that the homologous chromosomes line up side by side in the equator of the cell. Yes? Maybe this is your paternal chromosome, and this is your other paternal chromosome, and we have the spindle fibers going as such. The way that these two line up is independent of the way that these two line up, hence the independent assortment. Independent assortment of genes on different chromosomes occurs at metaphase 1 in meiosis. Is your eyeball okay? Good word. Alicia's flies didn't fly in there. What happens if a fly gets into your eyeball? See, you know.
It's eye drops.
Discomfort.
Well, discomfort. What does your body do? It destroys it.
Oh, so just like there.
Well, it ggets destroyed by your eosinophils. Those little infantrymen come over and they start dumping
I
hydrolytic
mean, like,
enzymes.
if it bathes up in your eyeball,
So it just
or
dies
like,
in your eye.
Yeah, it sucks to be.
well, it's like if it bathes up in your eyeball, or if it gets trapped in the socket.
Okay, anyways. The inheritance, independent assortment happens here at metaphase 2. The lining up of this chromosome is an independent event from the lining up of this chromosome. However, if you have two genes on the same chromosome, they'll be inherited together on meiosis 1. The only way that two genes on the same chromosome can be separated is via crossing over. Remember that in prophase 1 of meiosis, the two homologous chromosomes exchange corresponding parts. This is called crossing over. The two homologous chromosomes come together, they form a structure called a tetrad. Okay? And they overlap in a location called the chiasmata. And here, this exchange happens. In other words, crossing over would be like, if Rachel and Satya were crossing over, Rachel would like pop off her right arm, and Satya would pop off her right arm, and they'd exchange. So now Satya has like Rachel's right arm, and Rachel has like Satya's right arm. Crossing over. Corresponding parts of two homologous chromosomes swap. Does this make sense? This is like when you have like gummy bears, you do this in your room, you like pluck the head off of the gummy bear, pluck the head off from another gummy bear, and you switch their heads. Okay, linked genes do not assort independently because they're located on the same chromosome. Crossing over is the only source of recombination in linked genes. Check this out. The recombination frequency, this is your reader. The recombination frequency. It's given by the number of recombinant events over the total number of events. Now, let's say you had two genes that were perfectly unlinked on two separate chromosomes. What would be the recombination frequency? In other words, let's say the maternal genotype you got AB, and the paternal side you got AB. What are the chances of you getting this combination if you have non-linked genes? 50%, right? 50% of it will be that way, 50% of it will be this way. Yes? Does that make sense? If the maternal was AB and the paternal side was AB, then our recombinant genotypes would be AB and AB. And in unlinked genes, the recombination frequency is 50%. Yes? Now in linked genes, the frequency with which they recombine is less than 50%. Okay? Less than 50%. In fact, the closer two genes are to each other on the same chromosome, the less likely they will be to be separated. If you've ever been to Disneyland when you were young, your parents probably scolded you from wandering off too far. Yes? No? Because the farther you are, the more likely you are to be separated. Okay? Likewise, the recombination frequencies of genes can be used to construct this thing called a gene map. A gene map shows the relative loci. A locus is the location on the chromosome where the gene is. It shows the loci of different genes based on their recombination frequency. If we know that two genes recombine very frequently on the same chromosome, we know that they're very far apart. Because if two genes are here and here, crossing over can happen anywhere along this whole length, and you'll have the recombination frequency. You'll have the recombinant allele. However, if you have two genes that are very close by to each other, in order for them to be recombined, crossing over has to happen right there. Much more unlikely. Does that make sense? Any questions about that? Going once, going twice. Yes, no, maybe so. Now, I will give you this as an example. There's an anomaly here with this karyotype. This karyotype has 3 copies of chromosome 21. Okay? We call this aneuploidy. There are 2 possibilities. Well, there are multiple possibilities. We know what it means to be haploid and diploid, right? We need to review this one? We should. So if you're haploid, that means that you have 1 copy of the genes. If you're diploid, that means you have 2 homologous copies of the gene. Yes? What if you're polyploid? The ploidy describes the number of copies of a gene that you have. Okay? Haploid means that you only have N. You have 1 copy of the genes. If you're diploid, you have 2 copies of all the genes, 2 alleles. If you're triploid or tetraploid, has anybody ever been to Ranch 99 or like Asian supermarkets and they have like these huge Fuji apples? Crazy, huh? Plants are much more easily triploid than we are. If we get triploid, we don't at all. So these are called polyploid. Polyploidy is when you have an extra set. You have an extra set of genetic material. On the other hand, aneuploidy Aneuploidy means that we have an incorrect number of chromosomes. That is, we might have 2n plus 1. You might have 2n minus 1. You're missing a chromosome or you have an extra chromosome. That's aneuploidy. Aneuploidy means that you have an abnormal or incorrect number of chromosomes. Does that make sense? When we get to biotechnology, I will talk about polygenic, transgenic organisms and how we can use polyploidy to our advantage in genetic engineering. But for now, we just need to be familiar with the differences between polyploidy and aneuploidy. I have one quick announcement to make. Hold your horses. If you are taking the SAT Bio or SAT Chem, take one past that. If you're taking SAT Bio or SAT Chem and you need a 2-week like cram course,